This is a brief overview where I have divided my talk or slides under 4 sections. The first is measurement technology. The second is computing technology. The third is data science technology and functional allele testing. So in measurement technology, I will be talking about polymerase chain reaction, next generation sequencing, and application of sequencing. In computing technology, I'll discuss about the algorithms, like what you do when you have the large data sets, big data sets, efficiency, and what tools, basically computational biology softwares you could use to have your data analyzed. And third, that is data science technology. I will be talking about what you can do with the prediction, how you can predict the variation, like once you have the variant in hand, what would be this experimental design, correlation, and confoundings. I will not be talking about power, sample size, and causation, because that are mainly associated with complex diseases. But here my talk is more focused on rare Mendelian genetic diseases. And there I will take that an example, and then I will explain you how step by step we take approach for identifying the disease gene, and then what we do once we have the disease gene, then how we go for functional validation. And in the fourth section, I'll be talking about the functional allele testing. But before we begin, it is mandatory to know what is genome. So is anyone going to answer what is genome?
Genetic makeup.
Genetic makeup.
The genetic content of the organism can be defined as genome.
Yeah, wonderful. Great. So if we talk about genome, so let's understand what is the basic physical and functional unit of heredity. That is gene. So gene is composed of coding and non-coding segment, and that is arranged in chromosome. And there are 23 pairs of chromosomes that is arranged in the cell nucleus. And this all the DNA contained in a cell, or all the genetic information of a cell, is called genome. And when we say all the genetic information, we include mitochondrial DNA also, and the nucleus DNA that is organized in the chromosome, 23 pair of chromosome. So the central dogma is the explanation of the flow of genetic material. So within a biological system, starting by the DNA, which undergoes replication, which replicate itself to make a new copy, and then it undergoes transcription to make RNA molecule, and RNA, after processing and alternative splicing, gives you the protein of interest, or whether one protein or subset of proteins. It depends on what DNA is encoding or what information is present in the DNA. But if there is some mutation, or I should say genetic variation in the gene which is organized in the DNA structure, DNA, so what you would expect, the DNA will replicate with error. Either that genetic variation is repaired or not repaired. If it is repaired, it will continue to give you a normal copy. But if it is not repaired, then your replication will occur with error, followed by your transcription with error, and then followed by your translation with error, and then you get altered protein. And this altered protein, in terms of genetic, could have some loss of function, gain of function, nonsense mediated decay. So it can undergo these to any of these genetic mechanism. But we are not yet there. So we'll start with how, if you have this mutated copy, how you're going to know what is the mutation, what nucleotide is mutated, or how you can characterize the mutation. So before going on to understand how you're going to know, how you're going to characterize the mutation, let's discuss about the brief history of DNA sequencing. So in 1953, discovery of DNA structure was performed by Watson and Crick. The first sequence of 24 bases was published in 1973, followed by the Sanger sequencing method was published in 1977, and gene bank started in 1982. Then in 87, we have the first automated sequencer that was from Applied Biosystems Prism 373. And then in 1996, we have the first capillary sequencer. And from there, we have the first human genome sequence in 2000 to 2003. And in 2005, we have the first NGS sequencers that included Solexa, 454 Life Sciences, Antorin, and etc. So who is going to tell me what were the goals of Human Genome Project? Okay, so broadly, there were two goals of Human Genome Project. First, creating a physical map of the human genome, because till now we were not knowing what is the exact sequence in the human genome. When I say sequence, exact nucleotide. And then to sequence the whole genome of around 3 billion base pair. In addition to these broad goals, there were additional goals, and they were to make the information available for all researchers, thereby developing a computational system to store, transfer, process, and retrieve the data across the world, to identify all the disease-causing genes, to understand the function of gene, to map and tag genes on chromosome, to develop tools to process and analyze the data. So anyone could tell me why it is important to make the information available for all the researchers? The third point, can anyone explain why it was necessary to do that? Why this was considered as a goal? Am I audible?
Yes, ma'am.
Yes.
Oh, okay.
So you guys should respond and should interact with the presenter, okay? And if you guys not ask, I will ask everybody by name. I will call you and you have to answer them because it's very difficult to interact with you guys.
Okay, so understand the situation. If you're going to sit with your own data and you're not sharing with other people, for example, if you have identified a gene and you are not sharing that gene sequence or genetic information with anyone around the globe, how would you know that the gene that you have identified is actually a pathogenic gene? Does it make sense? If you're not sharing your why it is becoming important to share the information across all the researchers,
So that we can see all the online mutations. Matlab it is very important to share the data online because we can get to know what type of mutation is present and
yeah, you can get to know the pathogenicity of the variant. Then you could know how to transfer the files that you have identified, like in which you have identified your variant of interest, isn't it? And if you are dealing with the population genetics, so it becomes more interesting to see across the globe how your variations are present in your population versus other population, isn't it? Okay. So now moving on to the measurement technology. So in this section, I'll be talking about the polymerase chain reaction. So polymerase chain reaction is one of the molecular technique that is important to study the genome. And it is a technique that takes a specific sequence of DNA of a small amount and amplifies it to be used for further testing. And it's an in vitro technique. Can you amplify kind of like a whole genome through polymerase chain reaction? Why polymerase chain reaction was not considered while they were when they were just doing, why Sanger sequencing was considered? Okay, so what is the principle of polymerase chain reaction? The purpose, why we are doing it, condition, what is the condition to carry out the polymerase chain reaction, and what are the components that is required for polymerase chain reaction. So the purpose is to amplify a lot of double-stranded DNA molecule with same identical size and sequence by enzymatic method and cycling condition. So just imagine a condition of photocopy. Like with photocopy, you get one document printed copied in say 100 documents if you want. Similar, the like polymerase chain reaction also works in a similar fashion. What are the conditions? Denaturation is the first step of a double-stranded DNA template, annealing of primers, and extension of double-stranded DNA molecules. So what is denaturation? So the denaturation temperature is 92 to 94 degrees Celsius, where you can see in this diagram that this is a double helix, and at 92 to 94 degrees Celsius temperature, this becomes single-stranded DNA. So this process is called denaturation step. Then step number 2 comes annealing. The annealing temperature varies and it can range from 50 to 70 degrees depending on the melting temperature of the expected duplex. So in this case, the primers bind to their complementary sequences, forward and reverse primers, and then comes your extension. So extension, the temperature is usually around 72 degrees Celsius and the time is 0.5 to 3 minutes. And in this step, the DNA polymerase binds to the annealed primer and extends DNA at the 3' end of the chain where you get new DNA strands. So a cycle is composed of 3 steps, that is denaturation, annealing, and extension. I have used, here it is used elongation, but it is similar to extension. It is just another word for extension. So just to give you an overview, like an overall picture of principle of PCR, here the DNA has 1 copy, and when you are doing the PCR, you get lots of copy, as you can see in this slide. So what were the chemicals components used in carrying out the PCR reaction? So you can carry the magnesium chloride and their respective pHs, like molar concentration and pH is given, is written next to it. You have the buffer, you use TNTPs, ATGC, you have primers, the primers of interest, like primers designed for the gene of interest, which you would be interested in amplifying, DNA polymerase, and the target DNA. The target DNA, usually if you are amplifying a segment of the gene, for example, say 500 base pair, usually 50 nanogram per microliter works. But if you are dealing with more, say, if your downstream experiments are cloning, or say if you're dealing with, say, designing the plasmid, and in that case, you could go for 1 microgram. But in most, if you are just validating, if you're performing the Sanger validation, in that case, the target DNA could be around 15 nanogram per microliter. So you can dilute your DNA because DNA is precious. And what are the basic requirements for PCR reaction? So requirements for PCR reaction would be the DNA sequence of target region must be known. What you are going to amplify, that should be very clear to you. For example, in which exon it is present, and from where to from what starting point to what end point you have to amplify the region. That should be very clear in your mind. Second is primers. The primers are typically 20 to 30 bases in size. These can be readily used by commercial companies like Sigma, IDT, but also can be prepared using a DNA synthesizer. You need a thermostable DNA polymerase, that is Taq polymerase, which is not inactivated by heating to 95 degrees Celsius, because you want it to work in your annealing steps and extension steps. Basically, in extension steps, your first step is denaturation. And then DNA thermocyclers, this is a machine which can be programmed to carry out heating and cooling of sample over a number of cycles. So what happens if your PCR doesn't work? And that's mostly the great question, or mostly the problematic thing we actually come across when we are trying to amplify a region. So if no product of correct size is produced, try to check the DNA quality. Maybe your DNA quality is around 1.1 to 1.2. I have seen when your DNA quality is around 1.6 also, that works efficiently for amplifying the region. But if your quality is less than 1.6 also, then it may be a problem. So you can reduce the annealing temperature. Maybe you can fiddle around with the annealing temperature, couple of 0.5 degrees Celsius up or down. You can do that. You can increase the magnesium concentration, and you can add DMSO to say around 10%. And then you can use different thermostable enzyme. For example, if Taq polymerase is not working, you can use fusion Taqs or other Taqs which are high fidelity Taqs you can consider for amplifying your region of interest. Or maybe the primers that you ordered, when you were diluting the primers, you may have contamination. That's why you're not getting a product of interest of the correct size. So maybe you have to throw out our primers and get a new primer set, primer pairs. Or in the case if you have extra spurious product bands present, then what you would do, you would try to increase the annealing temperature, because you don't want unnecessary bands in your gel. You would try to reduce the magnesium chloride and reduce the number of cycles. Try maybe different enzyme. So this is an example of the PCR condition that you can start with when you're trying to amplify or when you're trying to begin your experiments. So first you can just have the initial denaturation at 95 for 5 minutes, and your thermocycle can be put it to 30 cycles of denaturation, that is 95 degrees Celsius for 30 seconds, annealing for 55 for 30 seconds, extension for 72 for 45 seconds, and then final extension for 72 for 5 minutes. And then you can hold the reaction at 4 degrees. What are the advantages of PCR? A small amount of DNA is required per test. You don't need to have, say, 3 to 4 microgram of DNA while doing those. While you're working with PCR, you can always dilute your DNA. As I told, 50 nanogram per microliter will be sufficient to amplify the region of interest. Then results obtained are more quickly. You can get the result within 1 day, or I should not say 1 day, also within, like, say 4 to 5 hours, depending on what type of you have, like the program you have. If you have like 2.5 hours or 3 hours programs, but so usually you get in like 5 to 6 hours, you get the results. PCR is much more precise in determining the sizes of the allele, especially essential for some disorders where you actually want to know what allele is present, what change caused the mutation. So you can, in that cases, PCR is much more precise. And PCR can be used to detect point mutation. So what are the application of PCR? It is molecular identification, like you can get to know about genotyping, prenatal diagnosis, mutation screening, genetic mapping, genetic matching, detection of pathogens, DNA fingerprinting. It can be used in sequencing. That is the basic principle for sequencing as well. Bioinformatic genomic cloning, human genome project, and genetic engineering, where you are dealing with site-directed mutagenesis, means you are making specific changes in the nucleotides in your sequence of interest. So, but what happens in the cases where you don't know what gene is causing the phenotype? You can use the PCR when your when you know what gene is causing, what gene is affected, what gene is causing the phenotype. But what we should do in the cases where we don't know what is the gene? So we should proceed on to next generation sequencing technologies, where first generation included Sanger sequencing, Maxim and Gilbert, and that based on Sanger chain termination. Second generation included 454, Solexa, and Torrent and Illuminate. And third generation, which is more common these days, is PacBio, or we should say long reads sequencing. So let's talk about Sanger sequencing. In Sanger sequencing, the DNA is fragmented, cloned through a plasmid vector, and that cycling reaction that is separated on the gel electrophoresis, and then we get the readout with the fluorescent tag. And let's talk about, whereas in whole exome sequencing, so why we want to perform the whole exome sequencing? The idea of performing the whole exome sequencing is when we have the individual with the phenotypes, and for that phenotypes, we are not able to predict the gene. We don't know what gene is actually causing the phenotypes. So in that case, and we first of all go for whole exome sequencing. And since the sequencing cost is declining day by day, so that is the preferred way of performing actually doing the genetic analysis in the clinic these days. So what is exome? Exome is the protein coding genome. So that is a split. It is a split among 160,000 exons. The total length is approximately 44 megabases, and it includes canonical splice size miRNA and exclude 5' and 3' untranslated regions. So, if here I have shown how what is the principle and what are the steps for next generation sequencing technology. You had a DNA. The DNA concentration is around 2 to 3 microgram, and that you just fragment the genomic DNA, then you ligate the adapters, build the library. There would be a cluster generation on the panels, and then they actually undergo the sequencing, and you get in the sequencer, which is like your Gemini High-Seq 4000. But why it is more efficient when it is compared to Sanger? So when we compare the NGS with the Sanger, sequencing of the human genome using Sanger technology took more than a decade, and it cost an estimated of $70 million. Whereas with the NGS now you can get the DNA sequenced in 3 days, and it is able to produce 1.68 into 10 raised to 9 bases. And this cost is also too much. You now you are getting the whole exome sequencing, I would say, in Indian money, like around 28,000 per exome. So, which is now the technology is actually now being spread out into all the, like, everyone can use the technology for the genetic diagnosis. So why we should consider whole exome as a and not the whole genome, isn't it? Because whole exome is just the 2% or say 1%, 1.5% of the total genome. Whereas if we just sequence the whole genome, we will be knowing about the coding versus non-coding. But why we don't go with that? It's because whole genome is still pretty expensive, and the pipeline or the annotation pipeline for whole genome is not well established, or it is still under the in the building phase, where you have like a lot of data and still they are like managing the pipeline for whole genome. Whereas in whole exome sequencing, the pipeline is established, and mostly it is like the human variations or disease is due to caused by the mutations in the exome, and it is significantly cheaper than sequencing the entire genome. So that's why it is more preferred in the clinic as opposed to whole genome. But if you have exome negative families, or if you have whole exome negative individual, then definitely you should consider whole genome sequencing in order to find out the reason or the variants behind the phenotypes that family or the individual is showing. So that's why whole exome sequencing is more preferred over the whole genome sequencing. And then finally, once you have the sequence data in the form of FASTQ or raw FASTQ form, you you have to perform a series of steps to systematically annotate the variants and identify the genes in the data sets that leads to specific phenotypes. In addition to whole exome and whole genome sequencing, I would like to introduce about the RNA sequencing as well. So RNA sequencing is also an next-generation sequencing technology in which you perform the transcriptomic analysis. So here you extract the RNA from the like cell of interest or the tissue of interest, and then once you have the RNA, in that RNA you have long non-coding RNA, small non-coding RNA, ribosomal RNA, poly-A RNA. Then based on what type of RNA sequencing you want to perform, you define the kind of whether you're going for poly-A selection. So this type of library preparation is actually going to select the RNA which has the poly-A tail. Whether you are going to ribodepletion, that means in this type of library preparation, ribosomal RNA will not be considered. Only long non-coding RNA and poly-A RNA will be considered. And then whether you're going for size selection, you only want the small non-coding RNA. So you would undergo for library preparation based on the specific library type you want in your experiment, and then you convert it to cDNA, and then you construct the sequencing library, and then followed by PCR amplification and sequencing. Similarly, like transcriptomic analysis, there is chip sequencing. In the chip sequencing, this is also a kind of next generation sequencing, where we want to see where identify the sequence or the nucleotides that are bound to the transcription factor. So in that case, we just do the sample fragmentation, perform the immunoprecipitation with the specific antibodies of interest, and then we do the DNA purification, do adapter ligation, and then sequence the reads. So what are the advantages of NGS? They can run in parallel, preparation can be automated, and it is much cheaper when compared to the traditional sequencing. So now coming on to the second section of my talk, that is computing technology and data science technology. So now you must be very much interested, like you started with a blood sample, and now how you can get to the segment of the gene that is actually causing the phenotypes from where you have the blood samples, or in the patient affected individual have the phenotypes. So what is the NGS pipeline? So we usually work with command lines. Why we work in command lines? Because it is easy to work with remote resources. You can just have SSH, you can SSH to your terminal, and you can log in if you have space in HPC's clusters, like high performance computing clusters. You can have your login over there. You can do SSH, type your username and to the login HPC account name, and then you can start working with your analysis. It is highly flexible to combine the small tools. So what do you mean by small tools? For example, if you are doing the whole exome sequencing, your first step is mapping, and then you have to carry out a series of data processing before you get the actual variant call format file. So you will able to combine the specific steps, and that in that case, the command line is very flexible. You can have a bash script, and you can have the looping of all the tools or all the software, so that you can have a idea, like you can get an idea, okay, from after mapping, you have to actually move the duplicates in the data sets. Then early availability of powerful and free programs. So you would definitely go with whatever is available free of cost. That you, for example, free, and it should not be only that. We should not be just saying like, oh, it's free, we can just use in our data. So it should be authentic and the validated, like data have been processed on these tools, because you don't want any, at the end of your analysis, you don't want to have any bugs that may be present in the program and that actually gave you different file as opposed to getting the actual file. The one drawback is that with the command lines, there is a limited visualization. There is visualization, but the tools for visualization are limited, and definitely you will have no distractions. So let's get started about the command line. If you have an Apple or Mac, you just open the terminal. If you have Linux, you just open the console or terminal extern, will open it. And if you have Windows, you can have just extern. The prompt bash commands are the best when you start with like when you have to talk about like, okay, you have say like you have one set of files that you want as an output file, but at the same time you want that output file to be the input file for the next step. So bash commands work at that interface very well. For example, over here I'll show like this sign send the output of a command not to screen but to file, but at the same time there is a double with the sign redirect the output and append it to the file. Whereas if you have the single line which creates the pipeline, that means after the output file of one command becomes the input file of the second command line. So that's how the command structures work. And if you're starting with say published tools or softwares, and if you need, like if you're stuck in between, then there is always a help or like help data also, which you can just query the tool. Like for example, if I'm using SAM tools for my whole exome sequencing, and if I'm stuck like while I was creating the SAM file from the BAM file from the reference genome, so I will just type SAM tool dash H. So that SAM tools will pop out all the scripts or bash scripts I could use in like fiddling around with my data sets. So there's always a help command, and LSH is a command line that you can mostly use for major computational biology softwares. So let's talk about the NGS pipeline. So you start with the unaligned BAM or FASTQ files. So what is the output file from your sequencer is a raw FASTQ files. Or sometimes some of the companies or say the labs give you BAM file as well. Then you use an aligner that is over here. I use Burroughs-Wheeler aligner, that is BWA, and to have my SAM file or either BAM file. If it is so, first of all, I have the FASTQ file. I will map it to the reference genome using the aligner, and once I map it to the reference genome, I will get the BAM file, that is binary aligned mapping file. So, what is the reference genome? Can anyone tell me? Have you seen like UCSC genome browser? You get SG19, SG38, or SG18. SG19, SG18. Okay, so when this human genome project was going on, when they started the human genome project, they started with sequencing couple of individuals. These individuals were not knowing why they are getting sequenced for, and they still don't know. No one knows. So this reference genome is not a single genome. First of all, it is a combination of individuals' genome, and the current reference genome has as less as I think is 250 gaps, whereas the starting reference genome was having 15,000 gaps. So why it is said reference? Because that particular genome we take into account and then we map our genome of interest. Like for example, if you're carrying out an association study, you have cases control. So you are comparing the phenotype of the disease with the control. If you are interested in knowing whether the disease or the SNP that you have identified, whether it makes the population susceptible to the specific disease or complex disease, for say cardiovascular disease, whether it makes yes or no. So you compare it with the control in that population. Similarly, you begin when you're doing the whole genome, whole exome, transcriptome, chip sequencing, any kind of NGS, next generation sequencing. Your first step is to map the sequence, because you want to know where it is different from the reference genome. And reference genome is not a single genome. It has multiple genome. It is like built up through multiple genomes, like around 13 to 14 individuals, but we don't know the ethnicity also. So then you map to the reference genome, then you remove the duplicates. So these duplicates were created while you were doing the library preparation and adapter, linker, addition, and everything on your like during the library construction. So Picard is the tool, is the software that removes the duplicates, and then we use GATK. GATK is another calling tool that do the indel realignment, that is insertion, deletion, realignment, and base quality recalibration means all the nucleotides, they will be calibrated about their base, like whether they have true positive versus false positive. So there will be a base quality calibration, and there will be haplotype caller, unified genotyper, and then we get the raw variant calling format. So here, now starting from say 4 gigabytes of file, we now come up with the file that is called raw variant calling format file, which contains not more, but around 60 to 70,000 variants in an Excel sheet. And from here, the actual genetic processing and actual work starts. Because from now raw VCF, you have to build up, you have to come to a VCF, which is just say 4 to 5 variants, or say maximum 10 variants. So that actually involves the role of hard filters, where hard filters would be, you remove the low quality sites. Low quality sites means the quality depth. If the nucleotide quality depth is like around less than 5, you will just remove it. If it has got allelic imbalance, that you see the data is considering the allelic imbalance, you'll remove it. So you do like, and kind of it's like fiddling with the data, where you kind of like you fiddle around with the annealing temperatures. Here you fiddle around with the data. You want to loosen your filters, you want to harden your filters in order to identify the variants that are pathogenic in the files. But once you have the variants, that also not enough. From there you want to go for that annotation, because still they are in the form of AAT and they are not linked to any, like what gene, like this variant belongs to which gene, you don't know where in the chromosome. Like, so you have to perform those specific parameters as well, where you will have the bill, and then you will just do the annotations, and then you will have the variant frequencies. So you will want the variant, you want to choose the variants since you are dealing with rare Mendelian genetics. So you will have the variants which have their frequency less than 0.001. Then you will see the conservation of the protein through different tools, like whether this particular gene is conserved, or this particular segment is conserved across different species. So you can use that PubMed for looking for conservation. Then for functional prediction of the protein, like if you have the variation, whether this variation is actually damaging versus benign in the at the protein level. So you will do functional prediction, and then finally you will do the gene annotation. Gene annotation means you will, whatever gene of interest you have identified through whole exome sequencing, you will put into the OMIM, and you will see whether you see additional, whether the gene has been published with those phenotypes or not, or it is a novel gene. Or then you would, if it is a novel gene, then definitely you would go and look for Jackson's laboratory knockout mouse models. So because in most cases, if you have a novel gene, then mostly the mouse is characterized. So you would get to know, okay, this is the mouse, knockout mouse, and the phenotypes of the mouse is neurological phenotypes or cardiac phenotypes. And the phenotype that we are observing in our patient is also neurodevelopment disorders, or say microcephaly, macrocephaly, and the mouse is also having neuro. So you will get an idea, okay, maybe based on the mouse model study, this gene is the top hit. But ultimately you will actually, when you will be actually be saying that this is the pathogenic gene, once you do the functional validation. So this is the end of a flow chart, where we start with genomic variants, say around 5 million, and they are like spread around chromosome 1 to 22 and chromosome X or Y. And then from there, we just select the variants which are high quality. And then we just we just move the variants which are common, that is present in population, more than 5% they are present, and we consider the variants which are rare. As I told that I usually take the frequency of 0.001 when I'm dealing with rare Mendelian genetics. And if I don't find anything at this frequency, then I go back and loosen my frequency. I go and loosen my filters, and I just start with, okay, let's see what variants I would see when it is 0.01. And then when you have these like frequency based file, then you would look for coding versus non-coding. Since we are dealing with exome sequencing data, so for the coding, we will have indels, nonsense, splicing, missense, synonymous. So indels is insertion, deletion. There may be an insertion of few sequences or deletion of few nucleotides. Nonsense, that means it may give it to the stop, like you may have a single nucleotide addition, or say 2 nucleotide addition that leads to creation of the stop or truncation of the at the protein level. And then you have the splicing. That means there may be variants that are present on splicing sites. splice donor and splice acceptor sites. Then there are missense, which is amino acid substitution, and then you have synonymous. That means they may not there may not be any change, but yeah, there's another story for synonymous, because now some synonymous actually give you, they may not be any amino acid change, but when they are actually clubbing and performing the function, they may actually affect the full function at the protein level. So missense could be damaging, and then you will make the final list of variants with the indels, nonsense, splice site, damaging, missense. And from here, you will actually perform, we will look for conservation, function prediction, gene annotation, and then you will be compressed to through an Excel sheet which contains only like around 5 to 10 variants, and most of the cases 20 variants also. But how do we integrate the whole genome, whole exome sequence data? So how many variants? That is difference from the reference sequence. An average of 11,000 variants per exome. And how do we interpret these variants? So first interpretation we do based on the pedigree, mode of inheritance basically. So if it is an autosomal recessive, for example, the phenotypes that we observed in the patient was coming from a consanguineous family. That is, there was, so in that case, we may think, okay, there is an autosomal recessive mode of inheritance. That means marriage between the cousins, and then you will have, so consanguineous family means marriage between the cousins, and then you have the mode of inheritance would be autosomal recessive. So in that case, you will tweak your exome data in that direction. You will only considering, or you will only extracting the genes which have the autosomal recessive mode of, which show autosomal recessive mode of inheritance. Then another would be autosomal dominant mode of inheritance, where one parent is affected, and then you will see the effect, the disease status passing on to their offsprings as well. There may be case of compound heterozygous, where you have a single gene having different, in the single gene having variation at two different exonic position. So that is called exon, and one exonic position is has the variant in mother, another exonic position as is present in father, and then the child inherited both the copies of the genes in an affected status. So that may be compound heterozygous state. There may be a condition where there would be a de novo, where none of the parents and mother and father are not affected, but the child is affected. So next generation sequencing is truly helpful in identifying the de novo dominant variations that was not possible before NGS, or that was really difficult to characterize, because for consanguineous families, one can perform the linkage studies, but for de novo, it was difficult. But it actually, next generation sequencing has already eased that difficulty. So as I told, like how to sort through extended variant list. Once you have the variants, say started like say around 50,000 or say more than that, how to start with that? So is the change, first of all, is the DNA change novel or known? So how you would get to know that? So the databases for that, for identification of that particular answer is DBSNP 1000 Genome Project or Exome Sequencing Project. These are all online tools that you can query on the website, and you can query your variation of interest in these specific websites. Then is the DNA change predicted to alter the function? So that prediction of mutation effects, whether it is coding or splicing change, or it is amino acid substitution. Does the DNA change segregate with disease? So in that case, you have to see the OMIM and human genomic mutation databases, and then you, after these particular filtering process, you get the candidate mutation. So progressive filtering is necessary to discard the artifacts, filter out benign variants, and identify pathogenic ones. So this is an example of variant gene list, where we have the nucleotide change, and we have the transcript ID, and we have the missense variant, and in addition to that, we have whether it is probably damaging variant or not, and what is its frequency in the population, and what is its CAD score that is allotted to all the variation, and the gene name. Through different annotation tools, that is through OMIM, HDMD databases, we came to conclusion, yes, this is the gene that causes the phenotype that we are identified in our patient. So NDFE1 was the gene. But once you have observed the gene, what next? You will go back, take out the samples of the individuals, and will perform the Sanger sequencing, because once you identify the gene of interest, you have to validate the variant also. And in this case, it was the autosomal recessive. So both mother and father were heterozygous. So over here, you can see there are two overlapping lines. Red is for T, blue is for A, whereas the proband was homozygous. Red is for T and blue is for C, and the proband was homozygous. So over here you can see a clean C, which shows that proband is carrying the homozygous allele. So now once we have the gene in hand and we have confirmed it through Sanger validation, what next? So first of all, if it is a novel gene, so we would like to share with the genetics community. So we will paste the gene on specific on some tools, that is like MyGene2 gene matcher, to identify additional variants, because then we will be more confirmed about our human genetics findings. Because if we have one patient showing up one gene X, but we still don't know whether one patient is not enough to show that, okay, this gene is actually causing the mutations that we have, phenotype that we are observing. So in that case, we want to have additional variants, and for that, to identify the additional variants, we will paste our variant on specific websites that are like, for example, Gene Matcher, MyGene2. If you're lucky enough and people are still finding the genes, the same gene, they have found it's the same gene, they will contact you, or you will contact to their collaborating physician, and you will say, hey, I have this gene and these are my patient phenotypes. Would you be interested in sharing your patient phenotypes? And let's see if we match on phenotypes and gene. Then if you're still lucky on, so you will actually find the additional variants and additional collaborating physicians. And from there, you can plan on to functional allele testing. So as I said, it is important, once you identify the gene, it is actually important also to functionally validate the variant that you have obtained through whole exome or genome analysis. So first of all, if it is a novel pathogenic coding variant, so if it is a novel pathogenic coding variant, then we want to have its genetic evidence and functional validation that is required for clinically actionable discovery. So genetic evidence, we have already shown that we have a gene that how we would be able to identify the genes through whole exome sequencing. And once we identify through whole exome sequencing, we can do Sanger validation. And once we do Sanger validation, then we move on to functional validation for mechanism of pathogenicity. And then the next would be the genetic evidence for impact of coding variant on gene. What is the variant type? Whether the variant type is truncating, missense, splice, nonsense mediated decay, or stably expressed. Where is the location of the variant in the gene? So when we talk about impact of the coding variant, it is more mainly on protein function. So whether it gives you disrupted function, necessary and sufficient, or introduce correct variation, and sanity checks, like does it make sense? So here I will show you an example of a CYT gene, that is citron kinase gene. So citron kinase, variation in citron kinase causes microcephaly. And there is a range of microcephaly severity. Some have moderate microcephaly, and some have severe microcephaly. So citron kinase is a genetic basis of microcephaly. So we identified in family A phenotype. Hello. Hello.
Yes, Anshika. Your
Yeah,
voice is clear.
okay. So in family A, we identified the splice variant that then we need to have genetic evidence and function validation. For that, we need to have additional variants. So for that purpose, we posted the citron kinase gene on the gene matcher, and then we were able to identify additional probands from gene matcher colleagues cohort. And so this was the identified cohort from the gene matcher, where family B was a consanguineous family, and there was a deletion of nucleotides, whereas in family C, there was a truncation of the protein. So then we have also identified additional variants, which was missense variants. And we observed that the missense citron kinase variant gives the phenotype of moderate microcephaly, whereas in case of truncating variants, it was more severe microcephaly, the very small brain, and the proband could not survive. As women they were born, they would die in a couple of days or maybe in few hours. So the genetic evidence was done through identifying additional probands through gene matcher. So this made us confident that the citron kinase gene that we identified in our patient is actually the causing gene for microcephaly, as we were able to identify additional physician with the same gene causing the similar phenotype, because microcephaly was common phenotype across the all the patients that had that harbors citron kinase variation. So next is that we have to do consistent genotype phenotype correlation. So for example, as I told, variant characteristics, whether it is a missense or truncating, zygosity versus clinical features. So for that, we made the we lay out the domain architecture of citron kinase, and then we mapped the mutations in the citron kinase domain. And we observed that the variations or the biallelic truncating variations associated with more severe phenotypes than missense, because we kind of correlated our genetic findings with our phenotypic findings. So in this, truncating missense compound heterozygous generates an intermediate phenotypes, whereas the missense were generating an if there is a compound heterozygous mutation, as I told, like if there is a citron kinase gene, and in citron kinase gene, exon 1 has a mutation and exon 4 has mutation. And if exon 1 mutation is carried in the mother and exon 4 mutation is carried in the father, and so in this case, the child will have both the mutations in citron kinase. That means citron kinase exon 1 and 4. So in the child, the variation will be present in exon 1 and exon 4, and the child will be affected, although parents will not be affected because they are having one functional copy of citron kinase. But in case of child, he have variation in both the alleles. So we proved it, we showed it that variant characteristics, missense versus missense truncating, and we showed that missense were having moderate microcephaly and the compound heterozygous were having intermediate phenotypes of microcephaly. So now we want to look at amino acid and protein domain versus clinical spectrum of phenotypes. So as I showed you over here, this is the domain architecture of citron kinase. And since all the variations are mapped into the kinase domain here, if you see, and then so it shows that autosomal recessive variants, they cluster in N-terminal kinase domain. So now we move on to functional validation, that is mechanism of pathogenicity. So if we start with functional validation, we start with impact of coding variant on gene. What type of variant is present? Is it a truncating, missense, splicing, nonsense mediated decay, stably expressed, location in the gene? So in our case, what type of variants were present? They were truncating, as you can see over here with this arrow, that these are the truncating variants. These are splice variants, exon 9 splice donor, and missense, where there is P158 to R, D230 to V. So these are the missense variants. Then we want to see whether these variants were undergoing nonsense mediated decay or stably expressed. So who is going to tell me nonsense mediated decay? What is nonsense mediated decay? No one? Okay, maybe anyone will tell me in the next class then. So the type of variants that were observed in this gene was all, that is truncating, missense, splice sites. Then we wanted to whether it is undergoing nonsense mediated decay or stably expressed. So we saw that it was undergoing nonsense mediated decay. So basically how we do it for the nonsense mediated decay. So if we have the mutation in the protein in the gene, then we will see and that mutation is a stop mutation. So definitely you will see the formation of mRNA will be there, but it will not be full length mRNA. Like the mRNA will not go for full length protein because of the mutation. You will have the stop before the actual stop. So in that case, if that particular mRNA is not adequate to the biological system, and if it is and when it is recognized by the biological system, then it undergoes decay mechanism. So over here, I am showing you, since we also identified the splice variant, so we wanted to show we wanted to perform the functional validation of splice variation. So for that purpose, first of all, we mapped or we just highlighted where the variation was present. And for that, we have a couple of sites, online tools we used. So this is the reference sequence, and this is the human splice finder, and this is the mutation splice finder. And in that reference sequence, and then this is the mutation sequence. So the 72.9 is the scoring of the nucleotide. That means at this site, it is 72% confident to have T at this region. This is the splice site. Similarly, we have over here in orange, there is the splice site, and then downstream, the starting of intron, the mutation was G2A. So here it is highlighted with disruptive difference donor site. And we wanted to see and we wanted to show how splice variant disrupt splicing. So in that case, we used mini gene assays. So what we do in mini gene assays, we have a vector, we take an empty vector, we amplify the region of interest, or in our case, it is citron kinase. So we amplify, and why exon 9 is taken? Because in exon 9, close to downstream of exon 9 was splice variant present. So we amplified the region of interest from wild type individual and cloned it into the PCAS2 vector. Similarly, we amplified the region of interest from the individual that harbors the variation. So we amplified that and cloned it into the PCAS2 vector. After performing the then we have like RT-PCR. To the RT-PCR, we were able to, and then it underwent to sequencing. And over here, the utility of a mini gene assay is we want to know which was the major and minor product. So if you are having the splice variation, you will either have retention of intron, or you may see deletion of exon. So once you have cloned that and then you perform the RT-PCR for your product and then you do Sanger validation, and then you would get to know whether some nucleotides were actually present from the intron in continuation to exon. So that is called retention of intron, or your exon was skipped. For example, in this case, it is exon 9. So then you run a gel and you see, okay, in wild type, you have a compact exon 9. But in mutant case, you have different products. So my major product was 4 nucleotide intronic retention, and the minor was exonic deletion. So with this, we say that citron kinase splice site variation disrupt splicing through retention of intron and not the depletion of exon, skipping of exon. So over here, we told that the impact of coding variation is we have the variant type all. We had seen truncating missense splicing variant, and we saw nonsense mediated decay, whether it was nonsense mediated decay or stably expressed. So we found that it was stably expressed because we are seeing retention of introns. And what is the location in the gene? So it is present in the kinase domain, because all the mutations that were identified in the respective families were clustered in the kinase domain. But what is, so with this, you show the variants that you have identified in a subset of families, that is citron kinase variant. You showed that they were missense truncating splicing variant, and you functionally showed that the splicing variant is actually disrupting the splicing event, and it is leading to retention of intronic exons. But what makes it more convincing, once you perform the functional assays, if there is a functional assays already reported for genes, then you can definitely undergo those functional assays, because in most of the cases when you identify the novel gene, mostly whatever information you get from model system, like for example mouse or zebrafish. It may have done or they may have created the knockout models, or some functional assays they may have done in, say, some cancer related studies or that. So you can do a quick function assess. For example, in this case, citron kinase, we were knowing that they are all present in the kinase domain, and we wanted to see the in vitro kinase activity. So we performed the kinase assay. So we cloned the human citron kinase in the flagged vector along with the and we also have the vector with the missense variant. And to the Kumasi blue, you can see that histone H1 was in vitro loaded, so that we can see the kinase activity in vitro. And you can see over here the kinase activity of the missense variants was similar to the empty vector, whereas it was not affected in case of full length citron kinase. So this is the normalized activity, mean density, where you have empty vector and full length, and you have the missense variance. So with all these introduction to genomic technologies, so now I'll come to an conclusion to my presentation today. So these all gives us implication for genomic medicine and basic science research. So if we talk about genetic professionals, this provides detailed understanding and supporting genetic evidence and functional validation that enhances the pathogenic variant classification. For example, once you analyze your whole exome sequencing dataset or whole genome dataset, you have a variant from the analysis, and through different tools and through different online websites, you process it through, and then you classify the variant, whether it is pathogenic or it is not pathogenic. So that gives a systematic approach. And about the research focus, once you understand what is the detailed understanding of the genetic mechanism, That means in this case, we saw that there were all kinds of variants. That means missense, splicing, and truncating variants. We saw in the humans, it was giving you brain phenotypes, that is neurodevelopmental phenotype, that means microcephaly. So when you know that the gene is stably expressed, so with all these information, this will lead to the selection of your model system. So that's why it is very important to have the detailed understanding of the genetic mechanism. Once you identify the gene underlying a specific disorder, it is very important to understand what is its genetic mechanism. Genetic mechanism includes its mRNA expression, its protein expression. Is there any functional assays that can explain what function it performs? As in this case, it was kinase function, phosphorylation. So on that basis, it informs the model system. So now from here, if you want to take on to understand how citron kinase is actually performing the function during early heart development, early brain development, because it is giving the microcephaly phenotypes. So now you can start it from there in your mouse models, if you're dealing with the mouse. So with this, I would, and these are some further reading, command line books and papers that you can consider reading while if you're interested in performing the analysis for whole exome, whole genome, or transcriptomic analysis.